So today we're going to be talking about muscle contraction. And before we get into the biochemistry of muscle contraction, let's take a second and just think about why animals move. Well, many organisms move in order to capture prey or to escape from predators. And one such predator is the cone snail. So cone snails are tropical marine organisms, and in terms of movement, they're actually fairly sluggish. But they're able to capture fish, which move relatively rapidly with a unique strategy. So they have a harpoon-like structure that injects fish with venom, and that venom leads to muscle paralysis. And once paralyzed, the fish is going to just get swallowed by the cone snail, and it really doesn't have a chance. If we think about the function of muscle in our own bodies, certainly muscle contraction is what enables you to get up off your chair, to walk across the room, to go for a jog. But muscle contraction is also what enables you to make facial expressions, to chew, to swallow, to breathe. So muscle function is critically important. There's individuals who have a disease called myasthenia gravis, and those individuals have impaired muscle function. So the individual in this picture has myasthenia gravis. You can see that the individual's eyelid is drooping, and it's drooping because normally there's muscles that hold it open, and in this individual, they're just not functioning properly. So by the end of this presentation, you should have a good understanding of how muscle contraction works. You should be able to, at the biochemical level, explain how the venom from the cone snail is leading to paralysis of fish. And you should also be able to explain how in myasthenia gravis, what's wrong at the molecular level that leads to impaired muscle function. Okay, so let's get into the details of muscle contraction. And the first topic that we'll cover just briefly is the structure of muscle cells. And muscle cells are packed full of proteins. And those proteins are critically important for muscle contraction. So we'll take a look at the proteins, and then we'll look at the mechanism by which those proteins work to generate muscle movement. And then finally, we'll take a look at how your muscles are controlled by the nervous system. Okay, so this is muscle tissue, and muscle tissue is made up of cells, and these cells are known as muscle fibers. And muscle fibers are kind of unusual cells because they're really long and they're multinucleate. If we look inside of muscle fibers, what we'll see is that they have many structures known as myofibrils. And myofibrils, there's about 1,000 myofibrils in a given muscle cell. And those are the structures that are composed of protein. And specifically, they have the proteins actin and myosin. So these proteins are going to work together to generate muscle contraction, and we have to look a little more closely at how these proteins are arranged in order to cause the muscle to contract. Okay, so the first protein we mentioned was actin, and actin is going to form what are known as thin filaments. While the myosin forms what are known as thick filaments. So there's a very orderly arrangement of these thin and thick filaments that we see here in this electron micrograph. So the thin and thick filaments are arranged here. You see that there's a repeating unit in the myofibril. So that one contractile unit that we see is referred to as a sarcomere. And we need to look at the structure of the sarcomere a little more closely. So what we're going to do is we're going to zoom into a cartoon diagram of one sarcomere that's present in a myofibril. Okay, so the top image is showing you one sarcomere in a relaxed muscle. And the bottom is showing you one sarcomere in a muscle that's contracted. So if we think back to our thin and thick filaments, the thin filaments in this picture are shown in green. And again, those are composed of actin. And the thick filaments are shown in purple. And those, we said, were made up of the protein myosin. And when you look at the thick filament, one thing that I want you to notice is it has these little heads that stick up off of the thick filament. And these myosin heads, their job is that they actually reach out and they contact the thin filament. And they're motor proteins, so they can actually move along the thin filament. And when they move, they're actually pulling the thin filament towards the center of the sarcomere. So actually, when a muscle contracts, the fibers or the filaments themselves, they're not actually changing in length. Instead, what's happening is the thin and thick filaments, there's an increase in the overlap of these filaments. So up here you can see there's not too much overlap, whereas down here the thin and thick filaments almost entirely overlap. Okay, so what we're going to do next is we're going to look at what's happening with this myosin protein head. And we'll look at the mechanism by which those myosin heads are leading to contraction of the sarcomere. So our first step is going to involve binding of ATP to the myosin head. And when ATP is bound to the myosin head, myosin is no longer going to make contact with the actin filament. So in step one, we've got ATP bound to the myosin head. And myosin, it is an enzyme, and specifically, it's a kind of enzyme known as an ATPase. So that means that it's an enzyme that hydrolyzes ATP into ADP, an inorganic phosphate. And you may also realize that muscle contraction is something that takes a lot of energy. So where is that energy coming from? Well, it's coming from ATP. So this ATP, it's involved in this mechanism, but realize that it's also providing the energy to make muscle contraction happen. Okay, so myosin is going to lead to hydrolysis of ATP, and the ADP and inorganic phosphate that are associated now with the myosin head, it leads to a change in shape in the myosin head. And these changes in shape in biology, we often refer to them as conformational changes. In this case, the myosin head cocks back, and with ADP and inorganic phosphate, the myosin head is also going to contact, in the third step, the actin filament. So the actin, this contact with the actin filament stimulates the myosin head to actually let go of ADP and inorganic phosphate. And when it lets go, there's yet another conformational change, and this one is known as the power stroke. So when this happens, the myosin head, you can see strokes like that, and that's going to move this filament in this direction, which will be towards the center of the sarcomere. So if the muscle were going to continue contracting, what would happen next is that the myosin head would once again bind to ATP, and that binding would cause the myosin to release from the actin filament. So one thing you may have heard of before is something called rigor mortis. When an individual dies, their body becomes very stiff for a period of time. And that stiffness happens because of the contact between the myosin and the actin. And an individual who's dead is no longer going to be able to generate ATP, which would normally bring these actin and myosin proteins apart. So with actin and myosin stuck together, it's going to leave the body very rigid until the proteins actually start to degenerate. Okay, so we've got the mechanism of muscle contraction. Now let's look at how the nervous system controls muscle contraction. So your muscles don't always contract. I can have my bicep relax, or I can decide to contract it. And the information for that is coming from the nervous system, and it's going to tell the muscle to contract. Okay, so muscles are then controlled by nerve cells. So this at the top represents one end of a motor neuron, and that motor neuron is directly adjacent to the muscle cells. And ultimately, the neuron is going to signal to the thin and thick filaments to generate muscle contraction. So how does this happen? Well, we have to take a closer look at the thin filament. So one thing we didn't point out before is that the thin filament has these darker green spots. And these darker green spots, they represent the myosin binding site. So that's where the myosin contacts the actin. The other thing we didn't point out are these orange proteins. So one of them is known as troponin. And there's another protein in orange called tropomyosin. So troponin and tropomyosin, they have the ability to slide down. So in a muscle that's relaxed, these proteins slide down, and what they do is they block all of these myosin binding sites, so that when the muscle's relaxed and the troponin and tropomyosin are on these myosin binding sites, the myosin can't make contact with the thin filament. And when muscle contraction occurs, it's occurring because the troponin and tropomyosin slide off of those myosin binding sites. And that is stimulated by the nerve cells. So let's go back and look at the neuron. And nerve impulses are going to travel to the end of this neuron. And when that happens, there's a few events. So one of the first events, when the nerve impulses reach this part of the neuron, it's going to open calcium channels. So calcium is going to move into the cytoplasm of the neuron. And that calcium in turn is going to cause vesicles containing acetylcholine to release that chemical, acetylcholine, onto the muscle cell. And the muscle cell has receptors for acetylcholine. And their response to acetylcholine inside the cell is to take calcium that's stored up in organelles and to move that calcium into the cytoplasm. And it's that calcium in the cytoplasm that affects troponin and tropomyosin. So that calcium, it's another one of these conformational changes. So the calcium associates with the troponin tropomyosin. It slides it off of the actin filament, and now the myosin head can contact the thin filament and will get muscle contraction. If there's no calcium around without the stimulation of a neuron, then the troponin tropomyosin will prevent muscle contraction. Okay, so going back to the example that we started with of the cone snail and its venom. The venom of the cone snail, when it gets into the fish, it actually binds to calcium channels in the motor neurons. And you can think of that component in the venom, which I'm drawing in green. It's like a plug. So it plugs up the calcium channel, and there's going to be no calcium that rushes into the cell. And without that, there will be no acetylcholine that contacts the muscle cell. There will in turn be no calcium, and the troponin and tropomyosin are going to stay complexed to the myosin binding sites, and muscle contraction will cease. And so the fish will be paralyzed. It turns out that the peptide that's in the venom that forms this plug of the calcium channel, we now call it prealt. And interestingly enough, if we're exposed to prealt, it actually doesn't lead to muscle paralysis. So our calcium channels look enough different from those of fish that they don't recognize prealt. Instead, the calcium channels that we have on special neurons called pain fibers, they do really resemble the calcium channels that are found in motor neurons in fish. So in fact, prealt has the ability to block these pain fibers, and it therefore is an effective painkiller. And in fact, its name stands for primary alternative to morphine because it doesn't have many of the side effects associated with morphine use. Okay, so lastly, let's take a look at myasthenia gravis. And this disorder, again, has to do with impairment of muscle contraction. And the impairment is due to impaired communication between the nerve cell and the muscle cell. So we said before that normally the nerve cell sends out acetylcholine, and the acetylcholine binds with receptors that are present on the muscle cell. So in myasthenia gravis, what happens is that the immune system kind of goes haywire. So normally your immune system is in place to help protect you from pathogens. And your immune system makes molecules or proteins known as antibodies. They're these Y-shaped proteins, and they're designed to attack things that are foreign to your body, like bacteria and viruses. But in some individuals, they end up making antibodies that are directed at self material. In this case specifically, they're directed at the acetylcholine receptor. So if an antibody binds to this acetylcholine receptor, well, now acetylcholine isn't going to be able to bind, and the nerve cell isn't going to be able to signal to the muscle cell to contract. Okay, so there are treatments for this disease, and the treatments focus on this enzyme here, which stands for acetylcholine esterase. So we have to think of a normal individual first. And in normal individuals, when acetylcholine is released into this space, it can't stay there forever, right? We don't want muscle contraction to be sustained. Instead, we have to clear that acetylcholine out. And that's the job of this acetylcholine esterase. So it takes acetylcholine and it breaks it down to clear out the acetylcholine from this area. So with individuals who have myasthenia gravis, there are medications that block the action of this of this enzyme. And what that's going to do is it's going to lead to a buildup of acetylcholine in this region between the nerve cell and the muscle cell. And if you think about the interaction of acetylcholine with its receptor or of this antibody with the acetylcholine receptor, these are transient interactions. And you can think of it as a war of concentrations. So if the concentration of acetylcholine is sort of on the low side and the antibody's concentration is on the high side, you're going to have more antibodies associated with acetylcholine receptors than acetylcholine. But if the concentration of acetylcholine increases, it can compete for binding with the acetylcholine receptor. And that buildup of acetylcholine that you get with medications that inhibit acetylcholine esterase, they're going to lead to better binding of acetylcholine with the receptor, which helps with the muscle impairment. So I hope that you've learned a lot about muscle contraction and that you see that our understanding of the biochemistry of muscle contraction has relevance to both disease and also to drug development.